Hello and welcome. My topic for today is combating COVID-19 with computational tools and strategies. On 31st December 2019, the Wuhan Municipal Health Commission reported several cases of pneumonia in Wuhan to the World Health Organization. And by March 11, 2020, it was declared a worldwide pandemic. The cause of these cases was previously unknown coronavirus, now known as the Severe Respiratory Syndrome Coronavirus 2, SARS-CoV-2, which can manifest itself in a disease named COVID-19. SARS-CoV-2 was an entirely novel pathogen, and in the light of the pandemic requiring a swift response to research and public health related questions, the natural first approach was to repurpose existing resources. This had a huge impact on all the existing virus bioinformatic tools that were applicable to coronaviruses and were then customized to address research questions related to SARS-CoV-2. In this video, we're going to cover bioinformatic workflows and tools that are helping in the study of coronavirus evolution, in the detection of the SARS-CoV-2 infection, the reliable analysis of sequencing data, and the tracking of the COVID-19 pandemic. In one of the first few studies on the new virus, the researchers were able to determine from the sequencing of the genome that the etiological agent was a single-stranded positive sense RNA virus of the Coronaviridae family. The bioinformatic tools for phylogenetic analysis revealed 89.1% similarity with the coronaviruses of the genus Beta coronavirus obtained from bats in China. The algorithm most widely used for comparing biological sequences, as we all know, is BLAST, Basic Local Alignment Search Tool. The diversity of information on biological sequences from organisms of different degrees of complexity allows BLAST to be a starting point to understand the genetic relationship with other species and also in the tracing of a possible origin. The use of such tool allowed to identify high similarity between SARS-CoV-2 genomic sequence that was extracted from NCBI GenBank with an accession number of MN908947 and the viral metagenomes found in the pangolin mammal present in the database. These studies in turn guided analysis that led to findings of high amino acid identity of the S, E, M and N protein of the pangolin coronavirus with those isolated from humans. This in turn suggested that the viruses that are capable of infecting humans may have emerged from a recombination of viruses isolated from bat and the pangolins. Moving on to the detection of SARS-CoV-2. The routine detection methods for the SARS-CoV-2 is a real-time quantitative reverse transcriptase polymerase chain reaction qRT-PCR. We know this, right? And you must have already started to wonder, how does this seemingly wet lab technique use bioinformatics or even a computer? Alright, let me explain. So the test is based on the detection of two nucleotide sequences, the virus envelope gene and the gene for RNA-dependent RNA polymerase. Specificity and sensitivity are the two most important quality criteria for the validity of any diagnostic test. To ensure unique identification of the SARS-CoV-2 and to avoid any false negatives and false positives, the computation of SARS-CoV-2 specific primers is required. A new set of primers might be required if the specificity and the sensitivity of the qRT-PCR changes due to mutations in the SARS-CoV-2 genome. And with so many strains detected right now, we know we need this. Talking about a tool that can help us design primers is PreSeti, the primer search tool. It is a software tool that identifies chemically suitable PCR primers in a reference dataset. Now, this reference dataset is nothing but a FASTA file of a complete genome or a set of short sequences of the genome. The diagrams on your screen shows you the SARS-CoV-2 specific primers computed by the PreSeti tool. and also the approximate amplicon locations of the de novo computed primer pairs for the virus, with no co-occurrences in any other genomes of the GenBank. Further on, talking about the genomic analysis. The RNA viruses exhibit huge amounts of genetic diversity within each individual patient. This genetic diversity is associated with viral pathogenesis, virulence, and even disease progression. Here, the schematic shows a single patient carrying varying genetic diversity over time. Next generation sequencing (NGS) is being used to assess the genomic diversity of the virus population in that individual. But NGS sequencing reads are typically short and error prone, and that is why we require its thorough analysis. Introducing VPipe. VPipe is a bioinformatics pipeline that integrates various computational tools for the genomic analysis of this viral intra-host populations. Most importantly, it supports the reconstruction of the genomic data of the viruses that were originally present in the population. Talking about how it works, VPipe takes input read data obtained from the viral sequencing experiments and produces in a single execution of the pipeline various output files covering the quality control, read alignment, and inference of the viral genomic diversity on the level of both single nucleotide variants and also viral haplotypes. But wait. Why does VPipe do all this? Studying the whole genomic diversity in the patient? Well, obviously, to improve clinical diagnostics and also to study the resistance of these viruses to certain drugs that can occur over time. And lastly, let's discuss a few things about epidemiology. Classical epidemiological models have seen a broad reuse in describing the COVID-19 pandemic. In brief, the SEIRD model involves individuals in a population that are grouped into susceptible, exposed, infected, recovered, and diseased. Initially, all the individuals except for the small number who are already affected are considered susceptible to that infection. The model can then simulate the population infection dynamics using criteria such as incubation time or average disease duration for parameterization of certain differential equations. Now, the first image shows the progression between these 5 epidemiological states that I just explained, with certain constants like beta, sigma, gamma, and so on. These in turn help formulate certain differential equations that represent the dynamics of the infection in our population. Using programming languages like Python, these differential equations are solved, and the solution helps plot graphs that can predict the progression of the disease. The animation here depicts how, with the infection-prone individuals rising to the maximum, there is a decrease in the susceptible population. And we can conveniently forecast the possible rise in cases in any particular area. Now, this seems easy, but we know we've gone through a lot. Extended versions of the SEIRD model were developed to guide even political decision making. For example, in Germany, this model is implemented in COVID-SIM, where they also included the hospitalized patients along with the patients under intensive care. Look at the convenient web interface this model has, permitting the user to change model parameters and get an intuitive feeling of the model dynamics, allowing it to estimate the infection parameters and to analyze the effects of contact reduction measures. Isn't that amazing? Here we have an intelligent machine elucidating a natural machine that brought the whole world to a standstill. These would just a few tools. And in case you're interested in exploring any of them, just click on these links and you'll be guided there. Thank you.